everyone. In this video, we're going to be discussing some of the peripheral endocrine organs and glands and the hormones that are released from each location in the body. So we'll start by discussing the thyroid gland. The thyroid gland is a component of the hypothalamic pituitary thyroid axis and is under direct control from the hypothalamus and anterior pituitary. So we'll just draw in the hypothalamus and anterior pituitary again, and then we'll draw the location of the thyroid gland. So the thyroid gland is anterior to the trachea and just inferior to the cricoid cartilage. So it actually sort of wraps around the trachea, and there's a right lobe and a left lobe, and it's connected by the middle piece, which is called an isthmus. And actually, when it wraps around the back, the location of the parathyroid glands is in the back as well. And on the next screen, we'll draw the parathyroid glands and take a look at those. So there are two basic types of cells that are located in the thyroid gland that are responsible for production of hormones. So the first major structure that is composed of the cells is the thyroid follicle. So it's basically like a spherical structure, and it's surrounded by the follicular cells or follicle cell. And these are actually epithelial type cells, and they just make a single layer. They're approximately cuboidal in shape. And so like epithelial cells, they will have an apical surface, which is the surface that faces the inside of the structure, and a basal surface that is connected to the basal lamina and in very close proximity to capillaries. So the thyroid follicle is responsible for the production of the thyroid hormones T3 and T4. And in order to make the thyroid hormones, we actually need a couple of important components. The first is iodide ions. And so the iodide is transported in the blood, and there are a couple of important transporters that are needed to move iodide into the follicular space. So the first transporter is the Nis Transporter. It's basically a co-transport mechanism for iodide ions and 2 sodium ions. And this is actually secondary active transport because of a sodium-potassium pump that's also in the same basal membrane. And so the iodide is transported into the cell. So it's actually up a gradient when it's moving into the cell. And then there is another transporter on the apical surface, which is actually called pendrin, which exchanges an iodide ion for a chloride ion, so just exchange of a negative charge for a negative charge. So the iodide accumulates inside of the follicle. Now, the other thing about these cells that's important to note, because they need to produce another important molecule, is that they have all the basic structures of a cell, including a nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, etc. One of the important chemicals that's produced by these cells is something called thyroglobulin. And thyroglobulin originates from tyrosine, which is one of the amino acids. The endoplasmic reticulum will synthesize thyroglobulin. It gets modified in the Golgi apparatus, and then it's transported into the follicle, the follicle as well. Once both of these components are present inside of the follicle, a cascade of reactions will occur that involves a series of enzymes that will ultimately produce T3 and T4. So the T3 and T4 will be synthesized, and they still actually are not quite finished because they have a thyroglobulin backbone still connected to them. But the structure that is complete is going to actually bind to some receptors on the apical surface of the cell. This is by way of receptor-mediated endocytosis. So simply, once enough T3 or T4 binds to these receptors, the membrane will pinch off and take in the T3 and T4 into a vesicle. Now, most of the time, the vesicle will fuse with a lysosome, and actually the lysosome will remove that extra piece of the T3 and T4, making it completely ready for export. Sometimes it will actually just move right through the cell. And what's exported from the cell, about 90% is T4 and about only 10% is T3. But as it turns out, T3 is the active hormone and T4 is the prohormone for T3. So even though only 10% is T3 initially, once the T4 arrives at peripheral tissues, it will be able to be converted to T3. Now, of course, we mentioned before that this whole process is under the control of TRH and TSH. So, of course, TRH is released from the hypothalamus, and it interacts with the anterior pituitary cells by way of second messenger. And then TSH is going to be released from the anterior pituitary, and that also interacts with thyroid cells in by way of second messenger. So the TSH will bind to a receptor on the basal surface of the cell after traveling through the bloodstream and cause a cascade of reactions that in turn will do a couple of important things. One, it will increase iodide uptake into the cell by increasing the number of Nis transporters, so the one in green. So the cell has the ability to synthesize more of those transporters, meaning making it easier to bring in more iodide ions. Also, it can increase the production of Thyroglobulin, also by way of interacting with the DNA. And then lastly, it can increase the endocytosis of the colloid being the T3 and T4 inside of the follicle. And so we could increase the number of receptors present and therefore increase the amount of T3 and T4 that's taken into the cell in a vesicle and therefore exported. If the hypothalamus or anterior pituitary is not recognizing enough T3 and T4 in circulation, we'll be able to undergo this process where we are then able to release more T3 and T4 into the bloodstream. Remember that T3 and T4 are actually lipid-soluble hormones, so they are going to be transported in the bloodstream bound to transporters, which means that most of the T3 and T4 will be bound, but there will be some that's free and therefore active. So the T3 and T4 travel to the target cells, and T4 is converted to T3 in target cells. So when what shows up as T4, it has to be converted to T3 in order to be utilized, but if it's T3, then it can be used right away. And once the T3 is inside of cells and in its active form, it basically interacts with mitochondrial and nuclear receptors to regulate gene expression. So this is going to be a common theme for a lot of these hormones, is that they interact with the nucleus and they can increase the number of proteins that are produced or decrease the number of proteins that are produced. Just to give you an idea of some of the cardiac effects, we can basically increase heart rate and stroke volume, therefore increasing cardiac output, increase systolic blood pressure, increase the number of beta-1 receptors that are present in cardiac muscle cells, and increase the ryanodine receptor associated with calcium channels, and inhibit the expression of NCX in calcium channels, which means the calcium would be available for a longer period of time. Also, it's responsible for affecting basal metabolic rate. When thyroid hormone binds to mitochondria, it will increase the rate of ATP production, which means that it will increase body heat and oxygen consumption. It increases glucose absorption and utilization, and potentiates epinephrine, norepinephrine, glucagon, cortisol, and growth hormone. So one thing that's really important to notice about these hormones is not only how many effects they have, because it's so many, but also the extent to which the hormones interact with each other and are either able to inhibit or potentiate the effects of other hormones. And we'll see that to be a common thread throughout many of these peripheral hormones. Now, of course, we mentioned already that inhibition of release of TRH and TSH occurs by adequate T3 and T4 concentration in the plasma. Other things that can inhibit TRH release include physical stress, starvation, and infection. So it's important to note that even though the hypothalamus is sort of king when it comes to making sure that the right number of hormones are circulating in the blood, other physiologic factors and emotional factors can affect the release of hormones, and sometimes we'll see that it inhibits the release, and sometimes we'll see that it increases the release, and in this case, a lot of these types of stress will inhibit the release. Alright, so now let's take a look at the posterior view of the thyroid gland and the parathyroid glands as well. As we mentioned, the posterior thyroid wraps around the trachea, and in the posterior aspect, we can also see 4 parathyroid glands in purple. The reason for considering these two structures together is because together they regulate plasma calcium. The other type of cell that's found in the thyroid gland is the parafollicular cell. And just to be clear, it's not only in the posterior thyroid. The entire structure of the thyroid contains both follicular cells and parafollicular cells, and they're in generally close proximity. And parafollicular cells are also called C cells, and they produce calcitonin. And the parathyroid gland contains chief cells that produce parathyroid hormone, which is abbreviated PTH. And the thing that's important to know about parathyroid hormone and calcitonin is that they actually work opposite of each other. They have opposite effects. Together, parathyroid hormone and calcitonin are responsible for regulating plasma calcium. So in plasma, calcium can exist in the ionized form, meaning just the calcium ion alone, and about 50% of the calcium is ionized. It can also exist bound to proteins, about 45% is bound to proteins, and it can be bound to anions, about 5% is bound to anions, and that would be things like phosphate ions, bicarb ions, and something else called citrate. We've already highlighted some of the important functions of calcium, such as its role in cardiac muscle contraction and in neurotransmission, communication between neurons, and some of the things that we're going to look ahead to is its role in skeletal muscle contraction as well as its responsibility in maintaining the integrity of bone. So generally speaking, if there is a disruption in the amount of plasma calcium, some of these functions could be dramatically impaired. And so these two hormones are responsible for sort of keeping the plasma calcium in check. So if there is an increase in plasma calcium, we'll get release of calcitonin. And it's been suggested that the role of calcitonin is sort of inferior to the role of parathyroid hormone in humans, but is a little bit more important in other animals. But in either case, release of calcitonin can lead to two important things. One would be increase of renal calcium excretion, so in the kidneys, more calcium will be filtered out and excreted. And two is inhibition of osteoclast activity. So osteoclasts are a specific type of cell that exist in bone that when broken down release calcium into the blood. So if their activity is inhibited, that means that less calcium would be broken down from bone and it won't contribute to the rising level of plasma calcium. Once the plasma calcium returns to normal, there will be no further release of calcitonin unless there was another rise. Conversely, if the plasma calcium were to decrease, the calcium sensing cells in the parathyroid gland will recognize that there is not enough calcium that is in the vicinity, and so we get release of parathyroid hormone, which leads to three things. One is a decrease in renal calcium excretion, so opposite of what happens with calcitonin. Also an increase in osteoclast activity, so more breaking down of bone to release calcium into the blood. And lastly, an additional function is to produce another hormone called calcitriol. And calcitriol is responsible for increased calcium absorption in the GI tract, so we can get more calcium if we need to. All these things will lead to a rise in plasma calcium back to normal. So as you can see, both of these follow a negative feedback pathway. We get a change from normal, recognition of the change, and then a response that involves release of a particular hormone. So this is the traditional physiological response-driven negative feedback. So different from the endocrine axis-driven negative feedback, we're seeing this sort of pattern. Alright, so I think that's enough for one video. In the next video, we'll move on and talk about the adrenal gland. Hope you enjoyed it. Thanks for watching.